All right, so it's my distinct pleasure to introduce our speaker. My distinct pleasure to introduce our speaker for Grand Rounds today, Dr. Jeff Pitt from Cornell, from New York. Dr. Pitt is the Ida and Theo Rossi Professor, Distinguished Professor of Medicine, and the head of the Cardiovascular Research Institute at Cornell. He really got started here at Yale, where he did his undergrad. I think was it biochemistry or classics. Okay. So he did his undergraduate degree here at Yale before he moved to Baltimore to do his MD and PhD training. And from Baltimore, he moved out west to Stanford to do his cardiology, his internal medicine training in cardiology, and did a postdoctoral fellowship with Dr. Dick Chen, where they really did some pioneering studies describing the regulation of calcium channels via calmodulin. From Stanford, he moved back to New York as an assistant professor at Columbia in pharmacology, where he established his independent laboratory that really has been at the forefront of our understanding of ion channel physiology in cardiac arrhythmias. From Columbia, he then moved to North Carolina, to Duke, where he rose through the ranks to full professor, establishing really, again, a very high-profile presence in the field of cardiac arrhythmias before moving back to New York, this time to Cornell, to lead the Cardiovascular Research Institute there. For those who are not familiar with Dr. Pitt's work, he's really been a pioneer in the study of cardiac arrhythmias, particular focus on ion channel physiology and biophysics, studying how sodium channels and calcium channels are regulated by upstream signaling, how their dysregulation can lead to a variety of arrhythmic disorders and other abnormalities, including neurological disorders. So we're really delighted to have you visit with us, Jeff. One of the last things I did at Mount Sinai was to invite you just before the pandemic, and so I'm really delighted to have you visit with us in person now. Now that hopefully the pandemic is behind us. I've said this before about your work, Jeff. Jeff is in our circles distinguished by a couple of things in his work. He's really distinguished with the rigor of his science, the way he does his science, which sets really the standard of how to study ion channels in the heart. And he's also distinguished with the innovation of his work, where he's really set so many different paradigms along the way. So, Jeff, we're again delighted to have you visit with us, and hope you enjoy yourself. Yeah.
I'm going to send your copy back to you. So thank you, Fai. That's a really nice, kind introduction, and it's really a pleasure to be here. As is the feedback from this microphone? Maybe I can turn off. I can turn this, I'm not going to wander. So maybe, does this work for everyone in the audience here on Zoom? I think it should be fine. But anyway, I want to thank you for the invitation. It's really a pleasure to be here. As Fadi mentioned, I perhaps got my start here on this campus, although I don't think as an undergraduate I ever made it over to the medical school. But there is a backstory to that. So my mom was a medical student here, and judging by looking at this room right here, maybe she actually had classes in this very room. I don't know where we are. So at least half of my alleles could have been here well over 60 years ago. But anyway, it's a pleasure to be here. And I figure since I'm returning to Yale, I was going to try to do a little bit different. So for those of you who are looking to hear about arrhythmias, there'll be a tiny little bit, but I'm going to try to go in a slightly different direction.
Sorry, maybe when I clicked on the pop-up there, it should go there.
Excellent.
Yeah.
Okay. So just by way of introduction, as Fadi mentioned, we're interested in ion channels and particularly in diseases that result from mutations in ion channels or in proteins that regulate ion channels. And so ion channels are expressed all over the body and cause diseases such as those illustrated here. Obviously, as a cardiologist, I'm particularly interested, as Fadi mentioned, in roles of ion channels in the heart, particularly arrhythmias, but other cardiovascular disorders as well. And lately, the lab has been able to begin to look at ion channels that are doing things in unexpected places and some unexpected things that they're doing as well. And that's been kind of fun. And I think that's what I'm going to try to focus on today. Obviously, excuse me, ion channels are great targets to look at because there are ready drugs that are sitting on the shelf that we can implement to treat diseases. And in fact, ion channels end up being the second largest class of proteins that are targeted by drugs after G protein coupled receptors. So it's a great way to translate our findings potentially into therapy. And in terms of this theme about looking at the roles of ion channels and learning lessons from what we've done in the heart and taking that to other places, I want to introduce to you Timothy syndrome. So this is a disorder that was described almost 20 years ago by Mark Keating, when at that time he was just kind of transitioning from Utah to back to Boston. And this is a multi-system disorder that's characterized by long QT syndrome. And so you can see a typical EKG here. And then patients with QT syndrome have such a prolonged QT interval that oftentimes these children are diagnosed in utero because they have 2-to-1 block. You can see that the QT interval's so prolonged here that you're getting 2 P waves for every QRS complex. And obviously it's this QT interval that then is the substrate for the arrhythmias from which many of these children die. It's a pretty rare disorder. And I think the reason for that is that if you look for variants on NOMAD, you find that there are almost none in the calcium channel. And thinking about arrhythmias in general, we have a lot of mutations in sodium channels, a lot of mutations in potassium channels. In the calcium channel, someone who's been studying calcium channels forever, I always say that the calcium channels are key to life because sodium channels and potassium channels are just setting the membrane potential to allow calcium to come in. And you tell me a cell and I'll tell you what calcium is doing. And obviously in the heart, it's leading to excitation, contraction, coupling. Despite the importance of the calcium channels, we have so few mutations in the calcium channels. And I think it's because it's so critical that the gene is just intolerant to mutations. But what's fascinating about this disorder is when it was first described as a long QT syndrome, there were several other features, one of which is invariant syndactyly, so fusion of the digits, hands and fingers, and oftentimes the children are diagnosed, they're born with syndactyly, they go for surgery, and then in the surgical setting, oftentimes after the fast, before the surgical procedure, they end up having their life-threatening arrhythmias. So how a calcium channel is leading to syndactyly is really a strange thing to consider and something that I've thought about for a long time, and I still don't have an answer. But there are several other features. There are neurological disorders such as autism. There are multiple hormonal disorders such as hypoglycemia that I'm going to talk a lot about today. And then there's some craniofacial abnormalities, all of which are the result of a single mutation in the calcium channel. And what I really want to highlight is the hypoglycemia. This is what I'm going to focus on today. And so I'll introduce that in a second. But just to kind of follow up on this theme, calcium channels are pretty much everywhere. And we're now seeing that they're also expressed in cells that are non-excitable, thus perhaps the syndactyly, still without a mechanism. And so for those of you who might be Lord of the Rings fan, I think that this is the one gene that does everything. But to kind of jump into what I want to talk about, this is the calcium channel, the cardiac calcium channel. The mutation is in the pore-forming subunit. It sits in the membrane. This would be outside. This would be inside. As I mentioned before, we're also interested in the auxiliary proteins that regulate ion channels. The calcium channels have both a beta subunit and this protein called alpha-2 delta subunit. The mutation is in the pore-forming alpha subunit, and it always sits right here. So all of these patients are unrelated, yet they have the same exact mutation in a specific residue. There are some rare patients that have slightly different mutations, but almost all of them have this specific glycine that's at the very end of the first set of 6 transmembrane domains. That's a glycine to arginine mutation. So this would be outside, this would be inside. This all folds up into an ion channel pore. And all of these unrelated patients have this particular mutation. Again, I think that as severe as a disorder as this is, I think this is probably a relatively mild mutation for the calcium channel based on the fact that we just don't see any other mutations. So this is a gain of function mutation. When we record calcium currents from channels that have this mutation, you see this lack of a process called inactivation. So this is a set of experiments where we're changing the membrane potential. And these are not our data, but these are the data from the original paper that we've done similar experiments many times. And each of these curves is looking at current, and by convention, positive ions, calcium going into the cells, a downward deflection. So this is current in the y-axis, downward current, calcium coming in, and in the x-axis, you have time. And you can see that calcium channels open relatively rapidly. And even while the membrane potential is held at a potential that's permissive for channel opening, you can see this rapid inactivation or channel closing properties. And what happens in patients that have the Timothy syndrome mutation is that closure does not happen as well. And there's 2 types of voltage or of inactivation for the calcium channels. This is a voltage dependent inactivation, which I'm not going to talk about today. But the net result is, because of this slow or ineffective closing, is that there's increased calcium influx, increased depolarizing current, longer QT interval, and then that's the substrate for the life-threatening arrhythmias. So now, as I mentioned, hypoglycemia turns out to be a severe consequence of this disorder, and it turns out to be a real problem for these patients. And it was something that I didn't actually appreciate when I first started looking at this disorder, mainly because of the role of the calcium channel and inactivation, which, as Fadi mentioned, is something that I had studied extensively, first as a postdoc and then when I was starting my own lab. But I had a chance to go to some family meetings and meet patients and their families. And many of these kids are having their arrhythmias in the setting of hypoglycemia or having hypoglycemia independent of their arrhythmias. And it's a real problem for these kids and the families. The parents are going to bed every night wondering whether their kid is actually going to wake up. And we had started developing some models, and we started to look at it. Now, the paradigm has been that, as I mentioned before, this is a gain of function mutation, so increased calcium influx. And so the paradigm has been that the hypoglycemia is driven by this increased calcium influx in the calcium channels that are expressed in the beta cell. So here is a review from, almost 20 years ago now, of the role of the calcium channels or the mechanism by which insulin is secreted from beta cells in the pancreas. And you can see focused right here is the voltage-gated calcium channel. And the thought was gain of function mutation, more calcium coming in, calcium triggers release of insulin from the beta cell, so a hyperinsulinism condition, and that's where the hypoglycemia occurs. And again, it's because of this inactivation process. Now, the hypoglycemia is really serious. Here is a group of patients that was reviewed about 5 years ago, and you can see the number of patients that I've highlighted in red that had hypoglycemia. And if you look at the number of patients that have hypoglycemia in the setting of their sudden death events, almost 3/4 of those cases can be ascribed to the hypoglycemia events. And as I mentioned before, it's oftentimes in surgery for syndactyly, which is a very common birth defect, that these patients then go for surgery. They fasted the night before, and during the procedure or right before the procedure, they can have these life-threatening events in the setting of severe hypoglycemia. So we had generated a mouse to do a bunch of other things. And then, as I mentioned before, after this patient meeting, we decided to look at the hypoglycemia. So I mentioned before, here's the mutation. It's interesting in that the mutation resides in an alternatively spliced, mutually exclusive exon, either exon 8 or exon 8A, that you can see here in code for almost the exact same amino acids. And the glycine that's mutated is the last amino acid in this exon. 2 of the nucleotides from that codon are part of this exon right here. And the patients are predominantly TS1, having mutations in exon 8A, although there also are patients that are TS2, which have mutations in exon 8. And the reason why I think that there are less exon 8 patients is because exon 8 is more highly expressed in the heart. And so there's going to be more arrhythmias. But anyway, we used CRISPR, we generated a knock-in mouse, and we were studying various features, and we continue to study various features. And we decided to turn our attentions to the hypoglycemia. And just to show you that when we make the CRISPR mutation and we knock it in, we can record from calcium channels where we know that this particular gene, CAC-NA1C or CAV1.2, the main calcium channel in cardiac myocytes, also expressed, as I mentioned, broadly. So here, for example, in neurons, and in red for the next several slides will be the mutant data, and blue is the wild type. And you can see the signature slow inactivation in isolated, in currents from isolated hippocampal neurons. Smooth muscle of the colon also express this gene. It's the only calcium channel essentially there. And again, you can see that this marked slowing of inactivation. The mice have long QT syndrome. When we stimulate them with intracardiac electrodes, we can trigger arrhythmias in the mutant mice, but not in the wild type mice. As with the children, they're sensitive to adrenergic stimulation. So here's isoproterenol, and you can see the arrhythmias in the mutant mice and not in the wild type mice. The mice survive, and we chose to study the TS2, which is the less common in humans, but it's more widely expressed. And mice survive arrhythmias, unlike humans, because their heart is so small, so they just don't have the mass to kind of maintain the reentrant circuits, and that's why atrial fibrillation is virtually impossible in a mouse, despite the thousands of papers that are published on it. But anyway, so let's go back to hypoglycemia. So the paradigm, as I mentioned, was calcium channels in the beta cells driving increased insulin secretion. And so that's what we wanted to focus on. And so first, and consistent with what I just showed you in terms of the cardiac features, which I'm not going to dive into today, the mice do show hypoglycemia. So this is a 10-hour fast. And again, you can see in red is the mutant, and they have low blood sugars during the entire time in the fast. When we do a glucose tolerance test, so this is the very similar test to what women get when they're pregnant, bolus of glucose, measure their glucose over time, although fortunately women don't have to have like 5 or 6 sticks in 2 hours as the mice have. but you can see that there's a decreased excursion of glucose in the mutant mice compared to the wild type mice. And so these mice with a single point mutation in the cardiac calcium channel gene, CACNA1C, are recapitulating the phenotypes of the hypoglycemia. And so here's where it got interesting. So we're expecting hyperinsulinism. That's what's published in every review. Although I will tell you, there's not been a single patient who's been described that actually has had their insulin measured and has been defined as having high insulin levels. So we measured insulin. So at baseline, and you can see already that the insulin is low. And then 30 minutes after a glucose bolus, when insulin should go up, and as you can see, it does go up in the wild type, it also goes up in the mutant, but it's suppressed in both. So this really kind of threw us for a loop, because now we have a mutation that we understand at the biophysical level very well. We had a physiology that we thought we understood very well, and we're getting the complete opposite results. So working with James Lowe, who's one of the other investigators in the Cardiovascular Research Institute, and is very interested in metabolism, obesity, as well as in beta cells. He helped us isolate islets, and then we looked at the function of the islets in different levels of glucose. And you can see that as glucose increases, you get more insulin secretion, but we see no difference between the wild type and the mutant. So in the setting in vivo, when we look at insulin in the blood, it's low. In the islets, it's perfectly normal. So we're essentially having two strikes now, and the results are kind of inconsistent. So we're really kind of struggling for what's going on here. So it certainly does not seem like the hyperinsulinism is the mechanism by which these patients, or certainly the mice, are having their disorders. And another clue is when we looked at the mice, they're smaller. And they have markedly reduced adipose deposits. So here are the adipose depots. Here's the epididymal and the subcutaneous. And you can see the wild type mice have what are typical looking adipose depots, and the mutants are about half of that. So a high insulin state should not have small adipose depots. You should, if anything, have large adipose depots, because that's a signal that there's fuel, and so you should be making fat. So again, this is not consistent with a high insulin state. Further, if you look at leptin levels, they're also reduced. Leptin is generated by signals from the fat, and again, it's reduced. And also in the fasted state, these mice have elevated ketones. None of this is consistent with a high insulin state. And so we were really confused. I really didn't believe these data for a while because it's so entrenched in the literature that we then generated another mouse model. We took advantage of a cDNA for either the wild type or the mutant calcium channel that's in a protected place in the genome in ROSA 26, and then it's silenced with a stop codon. And then we can use a CRE strategy to activate this gene in any specific tissues. So we used insulin promoter, and we drove the expression of this mutant channel solely in the beta cells. And then we measured the glucose excursion in a GTT, and you can see that it's completely normal. So even if when we drive expression of this mutant channel in the beta cells, we cannot reproduce the hypoglycemia phenotype. And then again, working with James, who recently obtained a single cell sequencing data set from beta cells, when we looked at which calcium channel is actually expressed in beta cells, you can see dark is higher expression. The related CACNA1D, which is a homologous channel, but different than the one that's mutated, is the one that's predominant in beta cells. So I think now we have an answer about why these patients are not having hyperinsulinism, because this channel is not even expressed in the beta cells, or at least expressed at extremely low levels. Physiologically, this actually ends up making sense because the CACNA1D, which encodes a protein called CAV1.3 compared to the CAV1.2 for the one that's mutated, that channel activates at more hyperpolarized potentials. And so it's sitting at a position to respond better to the action potentials that are present in the beta cells and therefore drive the insulin secretion. And if you look in human data from different data sets, you can again see, for example, in the beta cells that humans like mice also have predominantly the CACNA1D gene and not the CACNA1C gene. And that's pretty much true for almost all of the types of cells in the islets. There's a little bit of expression in the alpha cells, which I may come back to. So then what's the mechanism by which these patients and these mice are having hypoglycemia? Unlikely, but possible. We considered the possibility that they have glucose wasting. It's very unusual in the setting of low glucose because the proximal tubules are well tuned to take up glucose until patients are extremely hyperglycemic. There could be an abnormal counter-regulatory response, and there could be other mechanisms that were going on that we hadn't even considered. And so formally, not expecting this to be a mechanism, we actually took a look at the urine glucose, and you can see that urine glucose is actually elevated in the mice. And at the same time, their bicarb levels are reduced. And so this suggested to us that perhaps they have a renal tubular acidosis, which is a relatively rare condition in which there's either damage by toxins or by specific mutations to the proximal tubules. And so substances that are normally taken up in the proximal tubules are not. And that includes not only glucose, but also bicarb, showing why that there's increased glucose that's being secreted into the urine and decreased bicarb that's present in the blood. Patients that have renal tubular acidosis type 2 or Fanconi syndrome have also, they're polydipsic, and it turns out that we put these mice in metabolic cages that they're also polydipsic. And this is in the setting of normal renal function. So we think that there is a mechanism by which the calcium channels are potentially playing a role in the proximal tubules where these processes are happening. And the problem for us is that there's no data that suggests that the calcium channel is present in the proximal tubules. And so we have a reporter mouse in which the beta-galactosidase is knocked into the calcium channel locus. And so it allows us to use this as a reporter mouse, and we can see where the calcium channel is expressed. And so we used the mice, we looked at the kidneys, we can see it in the collecting ducts, which is the one place in the kidney where it had been previously reported using immunohistochemistry. But we also see it here, for example, in the proximal tubules, as well as in the glomerulus. And this was unexpected, but fortunate for us is just last fall, there was a laser capture, single sequencing data set that was published that says that actually it turns out that the calcium channel, this particular calcium channel gene is one of the markers for the cells in the proximal tubules. So these mice, kind of consistent with this multi-syndrome, Timothy syndrome, or multi-disorders Timothy syndrome, are teaching us new things about physiology. And so we now have a project in the future to look at, see what calcium channels are doing in the proximal tubules beyond what we're seeing here. I still don't think that that's what explains the main hypoglycemia, because the patients with Fanconi syndrome usually don't get such severe hypoglycemia. And so obviously the counter-regulatory responses were foremost in our mind to look at, particularly because the endocrine system is a system in which these calcium channels are expressed pretty broadly, as I mentioned before. And so we started looking at this. And so the first thing we did was an insulin tolerance test. This is similar to the glucose tolerance test, but rather giving a bolus of glucose, we now give insulin. And you can see that the wild type, after insulin, the blood glucose drops very quickly, as it should, and it begins to recover by 2 hours. And in the mutants, it drops and it essentially never recovers. In fact, in younger mice, and the patients that have the most severe hypoglycemia are younger patients, the mice died when we gave them the standard protocol insulin bolus. And we had to reduce the amount of insulin into a quarter of what we normally do in order to get them to survive. Because we were getting, glucoses of 10 or 20 and they would seize and the experiment would be over. And it was only after kind of backing off from the amount of insulin that we were able to complete these experiments. And then so knowing that insulin was driving the severe hypoglycemia, it suggested that there were problems with the counter-regulatory response. We started measuring all the hormones. As I mentioned before, the calcium channel is expressed in all of these endocrine systems. And we see that the corticosterone, which is the key counter-regulatory hormone rather than cortisol in mice, you can see that it goes up, and it also goes up in the mutant mice. So this seems to be intact. Epinephrine is also interesting. So the chromaffin cells are a cell that for years has been a cell to study the role of calcium channels. And so it's well known that they're expressed and play a role there. And so remember, this is a gain of function mutation. So again, in the wild type, after an insulin stimulus and a hypoglycemia stimulus, we see the appropriate increase in epinephrine from the chromaffin cells. And we see the same thing in the mutants. But notice that the mutants are already elevated at baseline and elevate even further in the setting of hypoglycemia. So I think that this gain of function in the chromaffin cells, in which calcium channels have a very well-established role, is driving a hyperadrenergic state. And I'll come back to that in a moment, because I think we have other evidence for that. The key hormone, as we all know, for the counter-regulatory response is glucagon, and that's, again, where we had a surprise. So with a hypoglycemic stimulus, you can see in the wild type, you get this marked increase in glucagon, and it's completely suppressed in the mutant animals. And when we isolated islets, doing the similar experiment to what we did for looking at the role of insulin secretion, but now at 2 different concentrations of glucose, is you can see that at high glucose, glucagon is suppressed, as it should be in the wild type, as well as in the mutant. And then when we lower the glucose, when we should get an increase in glucagon, you can see the marked blunting of the response in the mutant. So as an electrophysiologist, the next things that we're very interested in doing is recording from the alpha cells, as well as actually the delta cells. So somatostatin plays a key role in regulating these things within the islets. And so that's on the docket. I don't have those data to share today. But I want to come back for a moment. I mentioned before that these mice appear to be hyperadrenergic, or they have this kind of high sympathetic state. We think because the mutation in the chromaffin cells is leading to increased epinephrine release. And I think that also explains, at least in part, the decreased adipose depots in these mice, leading to increased lipolysis. So in addition to the hypoinsulinemic state, there's also this hyperadrenergic state. And so I think that's kind of consistent with what we're seeing. So now I mentioned we had this conflicting data. So in the blood, insulin was low. When we measured in the islets, that mechanism seemed to be intact. So neither was suggesting that it was hyperinsulinism, but it was also suggesting that there was a disconnect between what is happening in the isolated islets and what is happening in vivo. And this suggested to us that perhaps there was CNS regulation of glucose secretion. This has been something that has been a major focus for the past 10 or 15 years, looking at how particularly neurons within the hypothalamus are driving various aspects of regulation. And so using that same reporter mouse that I told you before, where beta-galactosidase is knocked into the calcium channel locus, we look to see where the calcium channel was expressed within the hypothalamus, which are the neurons that are critical to this glucose sensing. And we see that they're expressed in the arcuate nucleus, as well as in the paraventricular hypothalamus. These are 2 different regions that are critically important for glucose sensing. And we focus particularly on these initial studies in the arcuate nucleus. The arcuate nucleus has 2 major types of neurons. Ones that express pro-opiomelanocortin, or POMC, and the others that express AGRP or NPY, neuropeptide Y. And these are kind of like a yin and a yang. The POMC neurons are involved in appetite suppression. The AGRP, NPY neurons are involved in increasing appetite. And they appear to have different kind of opposite roles in a bunch of different metabolic processes, including glucose sensing. So because of this disconnect between what we saw with low insulin in the blood, but normal insulin secretion in the isolated islets, we thought that perhaps with this high expression in the arcuate nucleus, that perhaps if there were POMC neurons that were expressing the calcium channel. And these are all intermingled here. So there certainly are. And looking again at the availability of all the single cell sequencing data sets that are available, we can tell that it's prominently expressed in these POMC neurons. We took advantage of that to see that if we now drive expression of the mutant channel solely in these POMC neurons, using a Cree to release or to remove the stop codon from the cDNA that we have in this protected locus. So here's the genetic strategy here. Either the wild type or the mutant calcium channel is silenced by stop codon, and we use the Cree from that's driven by POMC to get rid of that stop codon. And now we express these specifically in the POMC neurons of the arcuate nucleus. And you can see that we can recapitulate that suppressed glucose tolerance test. And we think that this is specifically related to the inactivation properties, because if we drive the mutant channel, so both are slight overexpression, and we actually have a quantification of how much overexpression. It's not that much, but there is overexpression. But it's only when we express the mutant channel is that we see this suppressed glucose excursion. So we can recapitulate the glucose sensing when we express the mutant solely in the hypothalamus. And so we think that the CNS is driving this process. So let me check the time here. So let me start kind of with my first lesson from the heart. This is the sweetheart going from the glucose. So what we've been able to do is start from a cardiac arrhythmia, look at some of these other phenotypes, and learn some new things about calcium channels and physiology. We have a mouse model that recapitulates the key features of Timothy syndrome. We were able to show that the hypoglycemia is independent of the current paradigm, and it's not related to the calcium channels in the beta cells. In fact, we don't even think that the beta cells play a role because the calcium channel is not there. And then we have several other mechanisms that I've mentioned before, possibly a small contribution of glycosuria, and then particularly the blunted glucagon response, and then the role for CNS sensing. We're now doing some circuit physiology and setting up to do some optogenetics to really kind of trace these circuits within the hypothalamus. I think that's going to be really fun to continue to pursue. But to change gears slightly, as I said before, we've been able to use these models to look at a number of features of the Timothy syndrome and a number of features of the calcium channel, many of which had not been previously understood or well described. And I'm going to use this as an introduction to help us get us back a little bit into the heart. So one of the features of Timothy syndrome is that the children have a large mandible. And the mechanism for that was not that clear. And again, what a calcium channel is doing in the chondrocytes of the developing mandible to lead to this kind of hyperplastic response was unclear. But using the same model that I've now described several times is that we used a Cree that's expressed in these developing chondrocytes in the mandible, and we can recapitulate the phenotype. So this is one of the Timothy syndrome children who's now about 25 years old. I've had a chance to meet her several times. This is the picture from the original study. And if we drive the mutant channel shown here, but not the wild type channel in the presence of the Cree and only in the presence of the Cree, you can see that we can recapitulate here. The mandible is now extending beyond the maxilla. And in all of the other cases, all the other controls, no Cree or only the wild type channel, we don't see that. So this is a study that we did 10 years ago that kind of clued us into potential roles for calcium channels in chondrocytes and in osteoblasts. And we've done a little bit more. I'll show you one other study. We found that this could be a good mechanism or a model to study otosclerosis, which is kind of hardening of some of the bones in the ears that is sometimes present in patients as they age and can be very disabling because of problems with hearing as well as vestibular balances. And so, here is the beautiful apparatus in the ear. And if we look at the key ear bones, you can see that when we overexpress the calcium channel mutant, you can see how much thicker these bones are here than they are in the control animal. And the reason why we even started to look at these is that we had been expressing the channel in bone to look at some of the other features that I was just showing you in the previous slide. And I'll just replay that movie. And we noticed that these mice were spinning, so we thought they had some sort of a vestibular problem. And then we put them on this apparatus called a rotorod, which is essentially a way to kind of test how the mice can balance. And you can see that the wild type mice could stay on the rotorod essentially forever, and the mutant mice fell off very quickly, consistent with a vestibular problem. We were also able to measure their evoked auditory responses, and you can see that the threshold was markedly higher, so they needed much louder tones in the mutant mice than the wild type mice. So again, this could be a nice model for otosclerosis. But the reason why I'm bringing that up is there was an intriguing paper, and I also wanted to point out our Usain, who's sitting right here in the audience, who, when he was at Cornell, worked with Michael Matsui, who's one of the senior people in the lab on this project. And we had been intrigued by a study that had come out several years ago now that had identified this calcium channel that we've been studying and that's critical for Timothy syndrome, as well as a transcription factor that's critical for bone development, RUNX2, as associated with aortic stenosis. This audience being a cardiology audience that I don't always get a chance to talk to, I don't have to go over the details, but obviously this is the most common valvular abnormality, and explaining maybe about 3% of people when they get into their late 70s, and great for hospitals now that we have TAVR, they can make lots of money. So it's an important thing to study as well. And RUNX2 had previously been implicated in this process. The calcium channel had not. And so this study intrigued us. This study also pointed out that it was potentially a gain of function mutation, because when they looked at transcripts of several hits that they got from their underpowered GWAS and focusing on the calcium channel, you can see high expression is in yellow. You can see that in the disease samples that there was more expression of the calcium channel. So we have these mice, and let me just tell you one other thing, is when they looked at all of the SNPs that were associated with aortic stenosis in this study, and they put them into gene ontology pathways, that the very top was the calcium signaling pathway. So this gain of function mechanism, this increased calcium influx, increased signaling through the calcium channel, and we're sitting with these mice that have a gain of function mutation in the calcium channel in the same gene. And so we went down and looked at the oldest mice that we could find in our colony at the time. These were about 10 months old. And you can see that we have these features of this kind of chondrocyte-like appearance in the valves in the mutant mice and not in the wild type. And this actually looks quite similar to what is seen in histology of patients with early stage aortic stenosis. And so this encouraged us to look at this. And so this is where Usain started working with Maiko when he was still a resident at Cornell. And so they took isolated valve interstitial cells and put them into culture, and then used virus to transfect either the wild type or the mutant channel. And then we did RNA-seq to look at what genes are involved, but just to show you that by putting the channel in, that we can actually activate the process. So this is adding the Cree virus, and you can see that you get more stress fibers, more alizarin red, which is essentially a measure of the calcification process, increased smooth muscle actin expression. And this can be blunted if we then apply calcium channel blockers to the cultures. So it's suggested that the calcium channel, increased calcium influx through this channel, could be driving this process. We did, as I mentioned, RNA-seq. And if you look at the top pathways, again, we saw calcium signaling. Now, in our case, it's probably not a surprise because we're driving it by putting the calcium channel in. But in addition to calcium signaling, we're seeing signals of fibrosis, and we're seeing signals of bone formation here, osteoblasts, chondrocytes, and so on, only by expressing the calcium channel. So kind of consistent with what we think is going on in aortic stenosis. And then downstream signaling pathways for calcium, such as the NFAT pathway. And so we used, again, this model that I've now introduced several times. And this time we expressed either the wild type or the mutant channel, specifically in the valves using a scleraxis cree, which is highly expressed in tendons, and the mice end up with tendon issues as well. But it's also expressed in the aortic valve. And we can recapitulate these lesions when we express the mutant channel under control of the scleraxis, looking at these different stains. Von Casa, again, helps you with calcification, and alizarin red as a different way of looking at it. So when we express the calcium channel in the valves, we can recapitulate this process. We can see markers of chondrogenesis, such as aggrecan or SOX9, and we can see markers of osteogenesis, shown here, for example, RUNX2. And then all of this suggested to us that if the calcium channel is driving this process consistent with this GWAS, that perhaps treatment with calcium channel blockers early in the disease process could delay the development of aortic stenosis. And we were able to demonstrate that. So here are mice that we treated with relatively high doses of verapamil, starting at a relatively young age. And you can see that the lesion size is reduced in the animals that receive verapamil versus control animals. I'll also point out that these animals, the Timothy syndrome knock-in mice that I showed you in the first slide, are developing this on a normal chow diet. Most people who are studying aortic stenosis in mouse models will put them on a high-fat diet and use an LDL receptor knockout model to increase the lipid profile of these mice. And that's, and there are multiple studies that have recently implicated several genes that are involved, including the LDL receptor, that are involved in aortic stenosis. And our mice are developing this on a normal chow diet, suggesting that with this mutant calcium channel, that this is a really prominent feature. So calcium channels, as we know, we don't really use in aortic stenosis. We certainly don't want to use it in severe aortic stenosis. That would be a problem. But I don't think that there really be any contraindication to using these early. I've searched since we've had these data for someone who has a database looking at patients with aortic stenosis who have been on calcium channel blockers. Sometimes I always think that one of those great Scandinavian countries that monitors their patients so well would help me out, but I've never been able to find it. But I think that there are potentially supportive data. So ignored in the setting of coronary artery disease because of the strengths of statins, and now, as of a few weeks ago, bempedoic acid. We know that calcium channel blockers actually are pretty effective in reducing coronary artery disease. And so these are two studies from about just 15 years ago now, showing that calcium channel blockers can be effective at reducing the development of atherosclerosis. And because of some of the shared features between atherosclerosis and aortic stenosis, particularly around lipids, it suggests that calcium channel blockers might be early in the process an effective therapy. And it would take a good database to take a look at it. And I'm sure someone has the data out there. I just have never been able to find it. So how are we doing on time? Okay, so I have one more thing that I wanted to talk about because it also has a nice EL connection. Just a summary from this part is that we have these parallels between the osteogenic and chondrogenic processes by which the calcium channels are playing a role, such as the hyperplastic response in the mandible in the Timothy syndrome patients, allowing us now to move and look and see what's going on in the kind of osteogenic chondrogenic transformation that takes place in the development of aortic stenosis. And perhaps it hints that calcium channel blockers could be a potential therapy early in the disease process. So the last 2 or 3 minutes I want to talk about is the role of this other set of proteins that we've also been studying called fibroblast growth factors. So these fibroblast growth factor homologous factors. So this is a subset of the FGF superfamily. They're non-canonical because they don't get secreted. Even if one were to make recombinant protein and pour them onto cells that have FGF receptors, they have slight structural deviations that won't allow them to engage and activate the receptors. So they don't function as growth factors and they stay intracellular. No one knew what they did until in the early 2000s, Suliam Didhaj and Steve Waxman at their lab over at the VA identified these as interactors for sodium channels. And that really kind of got the field started. So this is the Yale connection. And around that time, we were trying to do structural studies, some crystallography of the calcium channels. And so the fact that these were interacting with the C-terminus of the sodium channel, which is an area that we were particularly interested in, is one of the things that got us interested. And I'm not going to talk about that part, but let me tell you that these are now known to be, they're very well established as regulators of voltage-gated sodium channels built on the work from the Waxman lab. And they've had several other roles that have been identified, such as stabilizing microtubules. In cancer cells, they appear to be involved in ribosomal biogenesis and regulation of protein synthesis. In humans, there are several diseases that have been associated with mutations in these genes. We identified some mutations in FGF12, which is the predominant fibroblast growth factor, homologous factor, or FHF, that's expressed in human heart, that's associated with Brugada syndrome. There have been various mutations in FGF13, which is the predominant gene that's expressed in mouse heart as well as in the brain, that are associated with seizure disorders. FGF14, there's a big New England Journal article maybe like 2 or 3 months ago. This is one of the major genes for spinocerebellar ataxia. And then something that I kind of wish I had inherited is that reduced FGF13 leads to hypertrichosis. I have the absolute opposite. The mechanism for that is not clear, but I think it would be an interesting thing to study. But let me focus particularly on the FHFs in the heart. As I mentioned, FGF12 is the main human heart FHF, and FGF13 is the one that's in the ventricle of mice. FGF12 is expressed in the atria. And I mentioned the cardiac arrhythmias. So here's the sodium channel. It looks a little bit like the calcium channel. The FHFs bind to the C-terminus in a region right near another protein that we studied that Fadi mentioned at the beginning, calmodulin, which is a calcium sensor. As I mentioned, we had used these to try to understand the structure of these proteins, and it worked quite well for that purpose. And no one had actually studied these in vivo in the heart. And so when we had done all of this crystallography, we built up a lot of tools and we decided to take advantage of this. Sure, I'm doing okay with time. And what we see is when we knocked down the FHF in isolated mouse ventricular myocytes, is that we saw a reduced current. So again, this is time in the x-axis, sodium positive ions going into the cell, downward direction in the y-axis. And you can see if we use a scrambled shRNA control compared to the targeted knockdown, as you can see that there's a reduced amount of current. So when we knock down the FHF, we see less sodium current. We're able to do biotinylation experiments to show that there's less protein getting to the membrane, which is one of the main reasons why there's less sodium current. And we think that's perhaps why we see, for example, in Brugada syndrome, that there are some mutations that are associated with it. Brugada syndrome, the main gene that's associated with it is loss of function mutations in the sodium channel. So here we have a loss of function mutation in an auxiliary subunit. It's also present in neurons. Depending on whether you knock down the FGF13 or the FGF14, you can actually get opposite results. And this clued us into the possibility that that this was regulating endocytosis, because the way that sodium channels get processed in neurons is they end up in an area called the axon initial segment. That's where action potentials begin. Highly concentrated sodium channels in the axon initial segment in neurons. And in the soma, in the main cell body, there's actually relatively few sodium channels. When they're generated, they go everywhere. They go to the soma, they go to the dendrites, and then they're selectively endocytosed from areas other than the axon initial segment. And so without going into the details, looking at the neurons clued us into what might be going on. But going back to the heart, if we now generated a knockout rather than a knockdown, we can see several features that are consistent with reduced sodium current, such as a decreased peak of the action potential. But we also saw a marked prolonging of the action potential. And that really was not consistent with a sodium channel defect. And we were able to show that, just as I showed you here previously in the knockdown experiments, focusing on the sodium channels, we're able to show that this prolongation of the action potential is related to effects on potassium channels. So we began to think that this protein is not only regulating sodium channels through its direct interaction, but is having other roles as well. In addition, we did kind of trying to think about what else this might be doing, is we did a immunoprecipitation and then mass spec to identify other proteins that are associated. And we identified these proteins called cavins. So cavins regulate caveoli. Caveoli have, again, a nice connection here at Yale, extensively studied in the vascular system here. When we knocked out the FGF13 in an inducible system and then subject these mice to transaortic constriction, we saw that they were protected from the heart failure that often happens after the hypertrophy that's associated with the increased LV pressure. And we're able to show that the number of caveoli were increased. And so what is happening is FGF13's acting as a negative regulator of caveoli. And when we knocked it out, there's more caveoli. And we think that there's just more membrane that's available as the cells hypertrophy to prevent them from developing failure. So we weren't satisfied with this as being the only mechanism. And so just in the last slide, I'll just mention that we've recently used proximity proteomics, building on experiments that were originally developed, or techniques that were developed from Alice Ting, where you have a biotin ligase that's then attached to a protein of interest. You express it in the cell of choice. Here we made a transgenic mouse. And then you use the biotin signals to do mass spec to identify proteins that are in the neighborhood. And so we identified about 700 proteins that were enriched, and we pulled out the cavins that we had previously identified, sodium channels and so on, alpha tubulins based on the data for the microtubules, a lot of stuff for the ribosomes. But then there were about 39 proteins that were all involved in endocytosis. So here's the KEGG pathway for endocytosis. And everywhere where there's purple, you can see is one of the FGF13 neighbors. And so, as I mentioned, we had been clued into the fact that this could be doing something in endocytosis. And it certainly appears to be that. We've now identified some of the key features that are part of that. And just to end, I'll show you that the lysosomal pathway is also part of this. If we put FGF13 and the sodium channel together now in a heterologous expression system, and we can drive the sodium channels into the lysosomes in the presence of the FGF13 is shown here in using LAMP1 as the marker, and you can see all these white spots. And then we, based on our crystal structure, we're able to design mutations that prevented the interaction of these proteins. So a single arginine mutated to an alanine in the FHF, or actually a human mutation at histidine that arginine sticks into the pocket, mutated to an arginine in patients that have arrhythmias. If we make either of those, we can prevent the sodium channels from getting into the lysosomes. And so here's the quantification of that here, and we end up with more protein. And so I'll just kind of end with one more picture of an MD PhD student who's now working on this project in the lab. noticed that within the data set that we had a bunch of intercalated disc proteins, and he was particularly interested in connexin 43. And so he's been exploring this, and when he knocks out FGF13 in the heart, he sees more connexin 43 protein, he sees more connexin 43 staining, and then the connexin 43 is not getting to the right spot. So here is looking at the interaction or the overlap with an intercalated disc marker and cadherin, and you can see that there's a markedly reduced overlap here. And then again, with this theme of endocytosis and targeting of the proteins to the membrane, he not only sees more protein overall, but less protein that's actually at the membrane when we do a biotinylation experiment. So just on time, I guess. So I hopefully went through some interesting non-cardiac as well as some cardiac features, and wanted to point out in pink are those people who were prominent in the hypoglycemia. I mentioned Maiko, she's driven this project. She also was the driver for the aortic stenosis project. Lauren was a former a technician who's now in graduate school who helped Maiko. I mentioned Yassine's work with Maiko, and then in blue are the key people that have done the work on the FHS, which is continuing. And then we had great collaboration from James Lowe's lab. So thanks for your attention, and any questions I'd be happy to answer. Yeah.
Fascinating talks, actually, in many areas. So I was intrigued by your finding in the CNS when you injected insulin, you actually have a lower glucose, which is suggests insulin sensitivity. I wonder if you have done example studies with these skeletal muscle, much more sensitive in this patient. If I don't say it because you have a low IUC, and normally this causes lipodystrophy and actually this study. And also, the one kind of suggestion, maybe you want to also look at the V1 creep and see if it's mandible is actually neuropressurized, but most facial. Think actually on neuropressurized, and maybe that's facial. That would be interesting. And also related to the same topic is that you showed actually benefit in aortic stenosis of the calcium antagonist. When you showed that the situation research paper, that also CACM-A1H that was changed, which is actually associated with hypertension, Gordon syndrome. So I wonder if that's actually your mouse has any representation for that mouse. That would be explaining the purpose.
Yeah, so let me see if I can remember all those things now. So in terms of the skeletal muscle, so we've looked at phospho-AKT and we've looked at AMPK, and we don't see any change in skeletal muscle in terms of insulin sensitivity. So the marked decrease in terms of the in the insulin tolerance test is still not exactly clear. I don't think that the glycosuria is explaining that. So we're still missing some part of that story. And I think that it could be something in terms of CNS regulation. There's now beautiful data that through the vagus that there's direct connections to the liver. And so we're setting up to investigate that.
So maybe you want to compare the oral injection.
Yeah. Yeah, so that's on the docket, as well as doing vagotomies and doing some other circuit stuff. So it's you know, we're not metabolic people, and so I'm kind of learning this on the fly, but it's a really cool area, and so I think we'll have some fun. In terms of the jaw, a lot of that is neural crest derived. The mandible in particular is develops through essentially chondrogenic like properties. We did some experiments with a zebrafish biologist. This is back when I was at Duke. And we couldn't get any real convincing data that calcium channels have a particular role in neural crest migration. But there are some data out there that are at least consistent with that. I probably forgot one thing, but yeah. Yeah.
Welcome back to Yale.
Yeah, thank you. Big one.
So your story, your demonstration maybe There's so many intracellular processes that are covered in makes me think that maybe many more of those cellular processes are governed by so that raises the question for me, because gain of function, that's self-explanatory, but what triggers in non-excitable cells, what triggers these capital? I don't think you talked about.
Yeah, so when we first started that was kind of one of the things that was kind of foremost in terms of what we were trying to explore. I think the definition of excitable versus non-excitable, first of all, is not really a great definition in terms of the way people understand it. There are a lot of cells that don't necessarily fire action potentials, but they still have a membrane potential, and that membrane potential can change. To me, the mountain to climb is to try to understand syndactyly. So that is a clear, because it's invariant in Timothy syndrome. And so the only mutation that these patients have is the mutation, this gain of function mutation in the calcium channel, and they're developing syndactyly. These are not, traditionally excitable cells. I would say one of my biggest disappointments in life is that our mice don't have syndactyly. And so are mice just different than humans, or is there some issue there? I don't know. We did some experiments in developing limb buds, and we could see in the mutant mice that there were, using a calcium dye, that there were different kind of like spike dynamics, but we weren't able to make really good progress there. So I don't know what the answer is, but it's a really cool question. Yeah. Yeah.
You mentioned the gain function and the besides the application, you observe
we haven't looked in terms of vasculature. We have done some work with the smooth muscle in the colon, because one of the issues that the Timothy syndrome patients have is a lot of constipation. And that's important because in the setting of kind of bearing down for a bowel movement, that has triggered arrhythmias in some patients. And the mice do have some issues in terms of their smooth muscle, but we haven't specifically looked into vasculature. So.
Yeah.
The CRISPR knock-in is global, so it recapitulates what the patients have. So they don't have sudden death, because it's very difficult for mice to have sudden death, but they do have, I showed you they have long QT. If we do intracardiac stimulation, we can trigger ventricular arrhythmias in them that we can't do in the wild type mice.
Is there change it and when
Yeah, so when we've looked at the arrhythmias and when we were doing that, we weren't focused so much on the hypoglycemia. I think what's happening in the patients is, as you point out, in iPS cells or in cardiac myocytes, if you manipulate the channel, it's clearly a role of the calcium channel in the cardiac myocytes. In patients, I think what's happening is the hypoglycemia is contributing to a hyperadrenergic state. And so that is then just making the arrhythmias that much worse. So I think that's the connection. So, okay. I don't know if there's any Zoom. It's a little bit over, but if there are any Zoom, someone.
Yeah,
tell me
Jeff,
how-
Jeff, it's Eric. If I can just
Oh,
make a
Eric.
comment. Hey,
Sure.
first of all, this is a tremendous work and congratulations. I just want to make a comment that, your work reinforces to me the importance of kind of the feedback loops that, genetics, physiology, basic and translational science and clinical science have to facilitate. I think we'll talk later, but I think the concept of using perhaps new computational methods to look at our large human phenotypic maps that are embedded within our EMR, for example, to look for associations between otosclerosis, and its downstream implications and calcific valve disease or oral maxillofacial surgery, and future events, I think is very, very intriguing. I think it's the kind of stuff that I think that's the weakest link, frankly, is the ability to kind of scan phenotypic features in large data sets that could help advance your science and lead to new findings. So anyway, congratulations, tremendous work, and we're very thankful that you were able to join us this morning.
Okay, thanks, Eric. Appreciate that. Okay, well, thank you, everyone.